Welcome to our webinar series for professionals.
Welcome everyone to this very exciting webinar, Mast Cells, Food Sensitivities and IBS, an insight with the GI-MAP test that we have so many practitioners using. So I know I have been looking forward to this webinar. When Tom suggested this topic to me earlier on this year, I was like, yeah, of course, bring it on. And the reason why that is because I just, in the clinic, I just see so many patients having these severe, weird symptoms where, well, the treatment isn't just a simple treatment solution. And I had a patient the other day where we had this chronic inflammatory response syndrome, CIRS, coming up, and also the mast cell activating syndrome came up, and like, it's just so complex. So it's going to be really nice to dig a little bit deeper into this area and hear Tom's experience. So anyway, I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life, and I am proud to be part of a global community with you all, where we as practitioners are dedicated to create health via functional medicine and personalized medicine. And I was actually just reflecting a little bit about all these webinars that we have, and I actually feel really, really honored that you all are joining in and signing up and asking for the recording and asking for the slides, which we will share, both the recording and the slides afterwards. So I think we've been able to gather, to create like this space where we as practitioners can ask questions during the talk, and I will answer as much as I can and bring the questions up at the end. And I think you bring a lot to these meetings, and also on our Facebook group when you ask questions and you help answering each other. It's not just us who answer. So I really feel that we all together have been able to create this online community. So I'm actually very honored for that. I just wanted to share that with you all. So, but today it's Tom Fabian who is the star of the show. And he has been here before. And it's 2 years ago or so that Tom was presenting with us last time. And I was just like reflecting a little bit over that. And then I thought, what would my boys say? So I have 2 children. One is 25 and the other one is 20. And I know the one who's 20, he would say legend. So that's where Tom is coming in here. He's been here before. He's done some excellent presentations. He knows a lot about the microbiome and health. He's a clinical laboratory consultant for at least for the GI-MAP test, and he is a translational science expert and a functional medicine practitioner. So he also knows how to work with patients, and so it's not just all in a lab or all through research. And he's a very good educator and speaker. So I'm excited to bring this next hour onto you all. So Tom, having said that, unmute yourself, please.
All right. Well, thank you so much, Anne-Catherine, for that great introduction. And thanks, everyone, for joining us today. I'm going to go ahead and share my screen here. And set it full screen. All right. So can you see the presentation okay?
Absolutely. Yes. Looks excellent.
Great. Okay. So let's go ahead and dive in. Obviously, the topic today is going to be focusing on especially the role of mast cells, the emerging role of these key immune cells in both food sensitivities as well as IBS. So we're really going to be looking at this full range of immune-mediated food reactions. And again, a lot of that is mediated by mast cells. So we're going to talk a lot about some of the research behind that. I kind of start off with a bit of a research synopsis. Then we're going to dive into essentially what do we look for on the GI-MAP? What are the imbalances that are related to food sensitivities and IBS, particularly both the overall picture, but really diving into the balance of the microbes in the microbiome. So we have, of course, the beneficial microbes, the opportunistic microbes, etc. So really identifying which are the ones that can stimulate mast cells, which can have immune effects in other ways. So we're going to really kind of dive in and sort of break that down quite a bit today. And if we have time, we are going to go through a case example. So again, these are the topics we're going to cover. So we're going to talk about the role of the microbiome, and again, particularly mast cells and histamine and food reactions. Really going to focus a lot initially on this new research. So we're kind of bringing the new perspective in to IBS and food sensitivities, and again, linking that with GI-MAP. And then we're going to talk a little bit about therapeutic implications, but mostly I'm going to save that for the Q&A. Because we have a lot of information that we want to cover. So I'll touch on that, but if you do have questions about how this applies therapeutically, I'm more than happy to answer questions about that. All right, so I'd like to start off with this sort of big picture focus on the typical traditional breakdown of adverse food reactions. So of course, we typically think of them as a combination of immune-mediated, so those are your IgE, food allergy type reactions. And then there's also a number of other types of reactions, cell-mediated. There are non-IgE mediated that are involved in things like celiac disease, other types of food sensitivities, and those are highlighted here. So we are going to talk a little bit about those today in the context of the overall picture, but we will touch on the non-immune mediated reactions, and those are, of course, generally thought of as food intolerances. And new research is starting to link both of those potentially to IBS, and also that there may be some common underlying interactions. So we tend to think of them as separate: food sensitivities, immune-mediated reactions, and then there's the food intolerances. So another kind of high-level approach that I want to talk about, or sort of perspective, is how clinicians typically address the scenario when patients may have immune-mediated food reactions, and what is the typical approach, and what is the research revealing where we can head to make our approaches more effective. So this first bullet point is really what a lot of clinicians start with, which is identifying the potentially problematic foods, and obviously then implementing an elimination diet. And that, of course, can be just sort of trial and error, starting with the most commonly reactive foods, gluten, dairy, etc., or through testing. Food sensitivity type testing. Really, though, what we're going to focus on is the second bullet point, which is all about identifying and addressing the GI imbalances that can underlie these food reactions. And that's really where there's a lot of hope that we can help patients reduce their overall sensitivity to foods and to other factors that they're exposed to. So that's really where a lot of the research is headed, because obviously patients that are highly reacting to a lot of foods, we really don't want to have a scenario where they're limited to so many foods, just a few foods in their diet. So let's talk a little bit about this first bullet point, just kind of walk through a typical scenario. So this is an IgG type test. Typical results, this is just one category. This is the milk and egg category. So this patient had just a few reactions to the egg white and egg yolk. You can see they're off to the right with the red dots indicating that there is significant reactivity. So these are fairly typical results for patients that have usually fairly limited food sensitivities. And then, of course, when patients eliminate foods based on food sensitivity testing, oftentimes that can help them reduce their symptoms. So there's a number of studies. The studies are sort of mixed depending on the type of test. But in general, IgG testing has been shown to help improve patient symptoms in many cases. So this particular test compared IgG testing to the FODMAP diet. So you can just see here from the title, I'm not going to go through this study at all, but just really highlighting this as just an example. It says IgG food antibody guided elimination rotation diet was more effective than the FODMAP diet and also the control diet in the treatment of women with mixed IBS results from an open label study. So again, just one study, but it does suggest that for some patients that the FODMAP diet may be appropriate For other patients, a more immune-mediated food scenario may be what's really going on for them. And it turns out the research that we're going to look at momentarily, really that can happen in IBS for patients where they have more of the immune-mediated or the more of the food intolerance type reactions. So this is kind of typical of the more unfortunate scenarios. So this is just the same test showing the same category for different patients where they were lighting up to everything. And we do see this occasionally where patients are reacting to a large number of foods. They know that from experience as well, that they seem to react to just about everything. So these patients are typically highly frustrated in terms of really relying on the clinician to help them understand what foods can they eat and then how can they reduce their sensitivities. That's really where getting into the bigger picture of immune-mediated food reactions can be quite helpful. So it's been known for quite a while that mast cells do play a role in classic food allergies. This is an example of a recent review talking about that. So that's really the sort of most well-established link between mast cells and food allergies. But again, there's this new research starting to broaden the spectrum beyond just food allergies and food sensitivities. So this study just came out. Actually, this is a review article of recent studies, I should clarify. So the title here is Immune Activation in IBS. What is the evidence? And I'm just going to highlight this section here where they say, in this review, we detail the current evidence suggesting that immune activation contributes to the pathology seen in patients with IBS and discuss the potential mechanisms involved. Moreover, we describe how immune mediators, particularly those released by mast cells, can directly activate or sensitize pain-transmitting nerves, leading to increased pain signaling and abdominal pain. So abdominal pain is one of the classic symptoms, of course, required for diagnosis of IBS. So there's a lot of research on understanding how is that happening. And really a long trail of research is pointing at mast cells as being key players in this sort of immune sensitization that can lead to abdominal pain. And it's often food related. So this is one of the actual studies published last year in Nature that really connected a lot of dots here. So I'll just talk a little bit about the study and then I'll focus on this excerpt here. But in this study, they showed that essentially evidence that we know that IBS, so that's previous knowledge, we know that IBS can be triggered by gastroenteritis or food poisoning, so a gut infection. So that's referred to as post-infectious IBS. There's been a lot of debates sort of among researchers as to what the actual mechanism may be. This is really one of the most compelling mechanisms that's been shown. And so they think that actually, at least for a subset of patients, that when they have gastroenteritis, that immune response, the inflammatory response, can predispose to sensitivity to a particular food that they may be eating during that time. So they actually can become sensitized to particular foods. And then later when they consume that food, that can stimulate the symptoms of irritable bowel syndrome. So that's kind of the summary behind this quote here, where they say, based on our data, we propose that IBS, at least in an immunogenetically susceptible subgroup of patients, is part of a spectrum of food-induced disorders mediated by mast cell activation, with systemic food allergy at the extreme end of the spectrum. So again, this is really kind of the newer perspective in research is IBS, at least a subset of IBS, probably a large subset, is part of the spectrum. So just kind of noting this here that probably all these sort of traditional sort of ways of breaking down food reactions, especially the immune-mediated food reactions, are likely to be revised as more and more research accumulates. But it really does explain a lot about that connection between previous GI infections, gastroenteritis, and then susceptibility to IBS and the involvement of mast cells and why patients react to foods and foods trigger their symptoms in IBS. I just want to touch on the non-immune-mediated part of this equation, primarily food intolerances. So food intolerances encompass a wide range of potential foods, of course, but the most common tend to be carbohydrate intolerance. as well as histamine intolerance. So we'll talk a bit about that. Now, this is from a recent review article that I would recommend if you haven't already come across this. This is titled Mast Cell Activation Syndrome, a primer for the gastroenterologist. And they say investigations of the effect of the low FODMAP diet in IBS-D patients have shown that there's actually a reduction in histamine levels as part of the diet. So, of course, with the FODMAP diet, we tend to think of it as being potentially effective for some patients by reducing their gas production. So that would reduce their symptoms of distension and bloating, potentially, and abdominal pain. But it turns out part of that picture may be also a reduction in histamine levels. And histamine has been shown to potentially play a role in IBS and visceral hypersensitivity as well. They go on to say, furthermore, high FODMAP diet in mice results in increased visceral hypersensitivity and increased mast cell density in the colon. So again, there's this connection now between, potential connection between carbohydrate intolerance, possible histamine issues, and then immune-mediated reactions. So you can imagine that for some patients, they may actually have reactivity both on the immune side, but also when they consume certain foods like high histamine foods, carbohydrates that like contain a lot of FODMAPs, et cetera, that may also increase their immune sensitivity. So this could kind of start to unravel some of that complexity with figuring out what's going on with IBS and then how to approach them from a diet standpoint. So a lot of this breaks down to, when you're talking about immune-mediated food reactions, the balance between oral tolerance, otherwise known as immune tolerance, and then immune-mediated food reactions. So once again, we're learning a lot more from research about this in recent years, and especially the role of the microbiome. So that's really going to be the main focus of what we talk about. And again, really understanding those imbalances and then knowing what to look for with GI testing is really key to helping clinicians understand what really should be focused on in order to help shift that balance so that patients may be more likely to have fewer reactions. And so of course, we know from research, this is just kind of a simplified schematic, that in terms of this oral tolerance, that's largely promoted by these beneficial commensal microbes. So key question with GI-MAP is, which are these microbes that we want to focus on and recognize when they're deficient? Then there's the pathogens and opportunists that can promote immune-mediated food reactions. There's actually a wide variety of them that have been characterized, and we're going to talk about several of these that are on GI-MAP. So this is a bit of a review for some of you that are more knowledgeable about sort of what's going on in food allergies, but I think it's a great summary, visual summary of sort of the contrast between what goes on in a healthy gut for patients that are not, or people that are not reacting to foods, and then also the classic scenario of food allergy, which is similar to what we're talking about with IBS. And this is from an article titled Regulation of Oral Tolerance by the Microbiome in Food Allergy. So I'm going to go through this figure in a little bit more detail here. So on the left, we have essentially the healthy scenario, where patients, of course, are consuming foods. They have a normal, healthy microbiome, producing these short-chain fatty acids that then promote overall health of the intestinal barrier, and as well as immune balance. And so you can see that down here, in particular with the immune cells. So this big sort of green cell in the middle of the highlighted area are the main cells that are characterized to promote immune tolerance. And those are the regulatory T cells, also known as Treg cells. But you can see that essentially one of the key roles of those cells is to block some of these other cells, the Th2 cells and the mast cells in particular, to keep them from overreacting. So that's really kind of a nice visual summary of what happens kind of at the core in a normal healthy scenario. You want to really promote these Treg cells through healthy diet that promotes healthy balance in the microbiome to continue to keep these cells from being overreactive. And patients can lose immune tolerance at any point. So this is an active process. You don't basically get immune tolerance when you're young and stay that way for the rest of your life. Some lucky people do, of course, but for many people, unfortunately, they can lose their immune tolerance because of gut infections, antibiotics, et cetera. On the food allergy side of this schematic, we can see here the food antigens are now in the context of dysbiosis and a dysfunctional intestinal barrier that may be featuring things like leaky gut, lack of secretory IgA, etc. Really, the consequence of all of that is then a disrupted immune balance, where now you have an increase in mast cells. They're primed to be reactive to food antigens, increase in these Th2 cells. So patients become more Th2 dominant. And then you can see in the middle here, it's just sort of summarizing this, that it's really all about the balance between the commensals and the dysbiotic microbes. And that's right here at the bottom of the figure. So key sort of advantage of using stool testing is that you can really get great insights into a lot of the factors that affect this balance. So you can basically get an assessment of the microbes that are right at the core of the imbalance, but also you can get insights into some of the other factors that are driving the imbalance, such as poor digestion. That's well documented that when patients don't digest well, that can increase their risk for food reactions. So let's talk a little bit more about these beneficial microbes. So again, this is from that same article that the figure is from, Regulation of Oral Immune Tolerance by the Microbiome in Food Allergy. They talk about that there are different immunoprotective commensal bacteria, including members of the Clostridia group, and also this large group within the Bacteroidetes phylum. So those large groups of beneficial bacteria that we know about quite a bit these days, and also that we have good representation on GI-MAP for, again, really go a long way towards promoting immune tolerance to these dietary antigens. So this is an example of a patient with a significantly deficient set of normal bacteria. You can see here there are a lot of lows that are flagged here, but especially at the phylum level. So that's the top taxonomic level. And that's essentially representative of 95% of the microbiome, depending on the patient. So sort of 90–95%. This patient was deficient in both of them. So this is a fairly common scenario. We see this in a lot of patients with chronic immune conditions, not just food allergies, food sensitivities. A subset of IBS patients will have this pattern. Patients with autoimmunity can have this pattern. So it's a fairly common pattern, and it's been well documented in research that lack of normal bacteria can predispose to a long list of chronic immune conditions, especially the butyrate producing bacteria. So that's primarily on GI-MAP, the Faecalibacterium prausnitzii, and that is one of the ones deficient here. And also, as we just saw in the previous slide, the Clostridia group. That's within Clostridia is within the Firmicutes phylum. So this would be a patient that if they do have known food sensitivities or food allergies or IBS, this could certainly be a big part of setting up the imbalance for this patient. All right, so this is a figure that I've used a lot in many of my presentations. It's starting to get a little outdated. It's 2014, but what it illustrates really clearly, that's why I like this figure, is just sort of the basic connection between a healthy diet containing plenty of fiber, and that fiber is metabolized by the commensal microbes to short-chain fatty acids. Those are there's many products that microbiome produces, but short-chain fatty acids are among the most abundant products and also the best well-studied. So you can see at the top here that essentially these short-chain fatty acids are stimulating the cells to produce mucus, also secretory IgA, and also tissue repair. So really important for the intestinal barrier. But also at the bottom, we can see that these short-chain fatty acids promote aspects of immune balance. So particularly on the left here, you can see here that these short-chain fatty acids promote the Treg cells and immune tolerance. And then they also can inhibit inflammation, particularly by acting on this particular well-known, well-characterized factor called NF-kappa B, that's a major regulator of inflammation. So it can inhibit that process. So several different ways in which short-chain fatty acids can promote a healthy barrier and also healthy immune balance. I'm going to talk a little bit about secretory IgA here. That is really kind of a connection I think that we want everyone to make is you can see this direct connection here between having a healthy level of commensal microbes that are producing healthy amounts of short-chain fatty acids in order to stimulate normal secretory IgA production. And that's important for immune tolerance or oral tolerance, as you can see here. I'm not going to go ahead and read through this, but you can just see this as an example of a review article summarizing the role of immunoglobulin A in oral tolerance. So if you see this low, really that implies a couple things. It implies that the microbes are likely deficient, or at least some key groups in the microbiome, such as the butyrate producers, are often deficient. And then patients may also be more susceptible, again, to food sensitivities, food allergies, and other immune type reactions. This is a common result. We don't necessarily see secretory IgA this low in many patients, below 100, but generally low secretory IgA is extremely common in patients where you also see a deficiency in the normal bacteria. So traditionally, clinicians have tended to think of this as maybe just a general immune deficiency and provide immune support, which can be certainly relevant for some patients. But really, because of that known connection now between low secretory IgA and low commensals, that's really where the therapeutic focus should be primarily in order to help increase the secretory IgA. Working on, and we'll talk a little bit about ways to support the beneficial bacteria. Also, short-chain fatty acids stimulate mucus production. So we don't have a direct marker for mucus production on GI-MAP. Mucus has also been shown, if it's deficient, to increase susceptibility to food sensitivities and food allergies. And you can see that summarized here in this review article. That's a missing piece of the puzzle in food allergy. So it's important to also make sure you're assessing things that can affect the mucus layer. So short-chain fatty acid producers, but especially, so that would be the Faecalibacterium that I have highlighted here. That's an especially important one. In this case, you can see this patient doesn't really have much in the way of overall deficiency, but these two species, which are known as keystone species, are commonly deficient in patients that have pretty significant barrier dysfunction, again, IBS, food allergies, etc. So Faecalibacterium is, again, a major butyrate producer. So that can have a negative effect on secretory IgA, and also the mucus production as well. But because Akkermansia depends on a sufficient mucus layer, that can be one of the reasons why it's deficient. That's often one of the major reasons. So when you see low Akkermansia plus other evidence of low beneficial bacteria, again, it's likely that that mucus layer is probably deficient. Of course, another key aspect of the barrier is permeability. So zonulin is a kind of an indirect marker for potential intestinal permeability. So it's a potential regulator of those tight junctions between cells. And again, we commonly see this elevated in patients that have particularly autoimmune conditions and allergic conditions. So patients that are highly reactive. And the other thing to think about with zonulin is it's sort of telling you that there's sort of this gateway is open so that imbalances in the gut can now be more likely to affect other parts of the body. So we're more likely to see patients with skin issues, joint issues, musculoskeletal issues, brain fog, mood disorders. These are all more likely, but especially food sensitivities and food allergies. We see this quite a bit in patients that have food reactions and IBS as well. So that's really a summary of some of the key beneficial microbes on GI-MAP and how to recognize that pattern, including low secretory IgA and zonulin. So let's talk a little bit about the key ways to support them. So we kind of like to summarize this as the 4 Ps. So most clinicians, I think, primarily are focused on probiotics when they see low beneficial bacteria, and that is important potentially for many patients. But really the key is all of these. So there's the prebiotics, i.e. dietary fiber, otherwise known as fermentable carbohydrates. Those are really the foundation for a healthy gut microbiome. And that's really what those really abundant commensal normal bacteria thrive on, and they need that to produce adequate short-chain fatty acids. Polyphenols is becoming more recognized as essentially having similar effects to prebiotics. They can promote the beneficial bacteria, but they have additional beneficial properties, such as, of course, being anti-inflammatory. So they can be very beneficial overall for gut health as well as the microbiome. Of course, probiotics, traditional ones, Lactobacillus bifidobacterium, spore-based probiotics, and now we have Akkermansia probiotic available. Unfortunately, it is a little bit expensive at this point. I'm hoping there's going to be more options available soon, so that price will come down. But it is potentially an option for patients that are deficient in Akkermansia. And then there's a new category. I think it's a little bit of a buzzword because really it's not a full category yet. But that's the idea of when you have a deficiency in beneficial bacteria and then a deficiency in their products like butyrate, that potentially supplying those beneficial products can still give you some of the benefit while you're working on increasing the normal bacteria. Of course, butyrate is sort of by far the most common currently, but there's a lot of research activity here to identify other beneficial compounds produced by the microbiome. So that's being referred to as postbiotics. There's quite a few studies out there looking at supplementing with dietary fiber and the effects of that in the food allergy realm. So you can see this is an article. I don't actually have the date noted on here. I think this was published 2 or 3 years ago. It's titled Dietary Fiber and Bacterial Short-Chain Fatty Acids Enhance Oral Tolerance and Protect Against Food Allergy Through Diverse Cellular Pathways. So there's just one of many examples of research that supports that increased dietary fiber can help to potentially reduce food allergy symptoms in some patients. Even before birth. So this is just an example of a study showing that the title here says prebiotic supplementation during gestation induces a tolerogenic environment and a protective microbiota in the offspring, potentially reducing food allergies. So really starting before birth with maternal diet can be quite important as well. We know that butyrate, which we just talked about, can actually inhibit mast cell activation. So once again, when patients have that highly reactive scenario, we know their mast cells are probably activated based on symptoms and potentially additional testing. Butyrate can potentially be both as a supplement, but also promoting butyrate production may be helpful as well. Interesting new research coming out on cannabinoids like CBD oil, and that they may also help to promote these T regulatory cells. There's some evidence now that that can also be helpful. Really, the research right now is not clinical so much yet. It's still kind of in the animal model phase, but there is some potential there. And we do know that cannabinoids can have some anti-inflammatory properties. This was a very interesting study that was done recently. So basically, they've really characterized what's going on in the small intestine. So the small intestine is the main site of food reactions. The large intestine, there can be some reactions if proteins are still intact to some extent by the time they get into the colon. They have documented reactivity in the colon as well, but primarily it happens in the small intestine. And it turns out that the small intestine permeability and immune tolerance changes on a circadian rhythm. So the long and the short of it is this quote here. I think this is really interesting and potentially something that's very useful for clinicians to know when they're working with patients and reducing their food reactions. So it says, the immune response in the small intestine was reported to have a specific rhythmicity, fluctuating between more or less tolerogenic phases. In light of these studies, intermittent fasting, with eating only during a restricted time period, might represent a novel strategy to prevent food allergy development or adverse food reactions. So this idea that, with a 24/7 lifestyle and people up late eating in the middle of the night, that may be setting the stage for an increased likelihood for food reactions. So restricting food to a certain period during the day when the immune system is more likely to be tolerant may help, again, to reduce those reactions. Now I want to talk a little bit about pathogens and opportunists, and the ones that have the best research on them, but also the ones that we tend to see a lot clinically with GI-MAP. That's a great review article from a couple of years ago, Mechanisms by which Microorganisms Influence Food Sensitivities. One mechanism through which bacteria could affect immune responses to dietary components is through bacterial metabolism of antigens. Indeed, depending on the type of bacteria present, the end result will be increased or reduced immunogenicity of the produced peptides. So we think, of course, of digestion as being the human digestive process, but bacteria, particularly in the small intestine, secrete enzymes that also break down foods, particularly proteases that break down proteins. So you have some that produce proteases that can create protein fragments that can be more antigenic, and then you have others that can break them down further so they're no longer antigenic. So we'll talk about a couple of those. Pseudomonas was identified as one of the ones that can actually promote food sensitivities, and also it's been shown to promote inflammation in the small intestine. So it's primarily a resident of the small intestine, even though we can detect it in stool because it's a DNA test. Says that Pseudomonas aeruginosa can metabolize gluten pre-digested by human proteases into shorter immunogenic peptides that permeate better through the barrier and then stimulate human gluten-specific T cells. And this actually is a pattern that we see on GI-MAP. So patients are more likely to have higher anti-gliadin if they have elevated Pseudomonas. So the research does seem to translate. And then when patients both remove gluten, of course, and reduce their Pseudomonas, then they do appear to be more likely to not react to gluten as much. So this is just an example of a patient that had fairly high levels. This opportunistic bacteria section on page 3 of GI-MAP is really where we see a lot of the evidence of these overgrowth type opportunists that are often involved in food reactions. Also, interestingly, there was a study from quite a while ago, about 11 years ago, that Pseudomonas aeruginosa was also linked to IBS. So this may be one of the mechanisms where it is linked to IBS. So really interesting connections now. Whereas lactobacillus, on the other hand, actually can protect against, in some cases, these types of food reactions and may actually help to specifically counteract the effects of Pseudomonas. They say here lactobacillus from the duodenum of non-celiac disease controls degraded gluten peptides produced by human and Pseudomonas aeruginosa proteases, reducing their immunogenicity. So again, that's how certain probiotics may be helpful, and also just building up the normal bacteria may be helpful. We often do see low lactobacillus in patients that tend to be reactive to foods. Just want to talk a little bit about Staph aureus. That's another one that we commonly see elevated in patients that have food reactions and common GI symptoms. So let's look at some of the research there. It's been long associated in the last, say, 5 or 10 years with not only skin issues, eczema, also respiratory issues, respiratory allergies, but in addition, it has been associated with food allergies and food sensitivities. So again, this is something that we commonly see. And we'll talk a little bit more about some of the research that can explain how that may be involved in IBS as well, if we have time. Candida is another one we commonly see elevated in patients that are reactive. Again, there's a growing amount of research on different ways in which Candida can affect the immune system, also has a negative effect on the intestinal barrier. This particular study showed that part of the cell wall of Candida can stimulate mast cell granulation, degranulation in the gut. There's actually an interplay among these. So oftentimes on GI-MAP on that page 3, you'll see high staph, high strep, high enterococcus, and then high Candida in patients that seem to have the worst symptoms. And there's some interactions here. We know from other studies that they can form biofilms together, particularly in the upper GI tract. And a lot of these are also oral microbiome residents. So if patients have oral dysbiosis, or if they have gum disease, periodontal disease, they may be more likely then to have these organisms higher in their intestine. But this particular one says interplay between Candida albicans and lactic acid bacteria which is primarily lactobacillus, enterococcus, and then also streptococcus. Impact on colonization resistance, microbial carriage, opportunistic infection, and host immunity. So Candida and lactic acid bacteria share metabolic niches throughout the GI tract. And in vitro studies have identified various interactions between these microbes. Two predominant lactic acid bacteria affected are lactobacillus species and enterococcus species. So the lactobacilli can antagonize these enterococcus species and also see Candida albicans, while the enterococcus faecalis and Candida have been reported to exhibit a mutualistic relationship. So they can kind of promote each other's growth, they can promote each other's virulence. So if you see both of those elevated in patients that have more significant reactions, it could be because, at least in part, because these are elevated. Research does support that. And again, we commonly see this type of scenario in patients that have a lot of reactivity. Another way to look at this is lots of studies now show which types of microbes in the gut can stimulate which types of immune cells and other inflammatory factors like cytokines. This is a summary review article of opportunists and pathogens that are known to interact with mast cells. To some extent, they can be beneficial because mast cells, part of their role normally is to actually help control these other opportunists from overgrowing. But if the balance is tipped too far, you don't have enough beneficial bacteria, you have too many of the bad bacteria and other microbes, then they may overgrow and excessively stimulate the immune system. So then you have more chronic mast cell activation. So the levels really do matter. If they're within normal ranges, they're less likely to cause mast cell activation. But quite a few organisms here. There's Staph aureus, Streptococcus, Pseudomonas once again, Enterococcus faecalis, Candida, and H. pylori. And these are all organisms that we commonly see elevated in patients that are reactive. Interestingly, there's also a connection between many of these and low stomach acid. So lots and lots of studies, enough that they can do these systematic reviews, have shown, and this one mentions that they reviewed 19 eligible studies. So these are studies that met certain quality criteria, and they consistently found high strep, higher enterococcus, higher staph, and bacillus. So that can be one way to reduce these organisms, just making sure patients are digesting well, producing enough stomach acid. Interestingly, also, there's research showing that acid suppressant medications can increase the risk of allergic diseases. Just reading a couple highlights here in summary, exposure to these medications prenatally in childhood and adulthood may increase the risk of allergic diseases. And then just sort of their summary is that there's a growing body of evidence to support that exposure to acid suppressant medications can increase the risk of allergic diseases. So again, it may be through causing these mast cell promoting type organisms, promoting their growth in the gut. And I want to talk just a little bit about the role of histamine and histamine producers. So Morganella has been widely characterized as a common, potentially high histamine producing microbe that may contribute to histamine symptoms for some patients. This is from a recent review on histamine intolerance, just highlighting Morganella and Klebsiella pneumoniae among the ones that are the most prolific histamine-forming bacteria. Interestingly, there's a connection between histamine and mast cells, and not only the traditional connection where we know mast cells can produce histamine, but as we can see in this quote, summarizing the research, that histamine can stimulate mast cells. And that's the highlighted part, that mast cells not only release histamine, but the mast cells have receptors for histamine, so they can be stimulated by histamine. So you can start to see, once you have an imbalance of these microbes that are stimulating mast cells that are producing histamine, that can set the stage potentially for increased reactivity. This is a list of the histamine-producing bacteria that are known from research on GI-MAP. Many of these we do often see elevated in patients that have outright histamine intolerance, but also patients that just have more subtle potential connections like food sensitivities, et cetera. So it's good to know this list. We should have the full list on our website sometime in the next few weeks or so. So Anne-Catherine, I can kind of give you a heads up and send you a link if you'd like to share that at some point.
Yes, please.
I'd be happy to. So that's really kind of the summary of some of the key pathogens and opportunists on GI-MAP that can promote immune-mediated food reactions in various ways. Just want to touch briefly on, of course, we all know that most clinicians tend to use antimicrobials. It's kind of their go-to, different protocols for different microbes, whether it's Candida, et cetera. But being aware of additional ways to inhibit those microbes can be really beneficial. So earlier we talked about polyphenols, for example, not only help promote the good guys, but also help to reduce the opportunists. And once again, lactic acid bacteria, that's primarily lactobacillus in terms of probiotics, protect human intestinal epithelial cells from Staph aureus and Pseudomonas aeruginosa. So once again, this is where certain probiotics may be helpful for certain patients, and also fermented foods. One of the advantages of fermented foods is that oftentimes they'll contain significant lactate. So the active factor, typically, there can be other factors, but the main one tends to be the lactic acid itself. So additionally, fermented foods may have additional benefit beyond just probiotics. Apple cider vinegar. So we just talked about the acidity and how lack of adequate stomach acidity can result in an overgrowth of some of these microbes. But even just simple apple cider vinegar has been shown to inhibit some of these microbes as well. And that may be one of the ways in which apple cider vinegar has beneficial effects. So hopefully you can see through what we've talked about so far, that what we can basically detect and analyze on GI-MAP really includes a lot of the key microbes and processes that we can recognize for various patients, and then that helps us connect the dots along with this type of information coming from research so that we can really understand what may be going on for patients, and then also how to target that. So Anne-Catherine, do we want to go to questions now, or would you like me to go through the case example?
There are a few quick questions, and then we can do the case. Let's try and do that.
Great.
Do it that way. The first question that came up was, and that's just, I'm also curious just to hear what your answer is in regards to food intolerance testing. For how long time should people be eating the foods before we can be sure that the test is correctly positive or correctly negative?
So for IgG type, is that what you're asking about, IgG type testing?
Yeah.
IgG testing, generally it's going to vary a bit from patient to patient and what their levels are, but the idea is that you want to test them within ideally a month of having consumed the food, maybe as far out as 3 months for some patients. So for anti-gliadin on our test, the GI-MAP test, we can still see that elevated for up to 6 months after patients have stopped consuming gluten. Generally, it will start coming down, but it may still be high for some patients, so it can take a while. IgE is a little bit different than IgG. So IgE is more short-lived. So if you're doing a food allergy test, the idea is that you need to be exposed to that typically within the previous few weeks before the test to increase the chance that it'll show up as positive. So a little bit of a difference between the timeframes between the two types of testing.
Then another question came up in regards to, like, of course, increasing short-chain fatty acids with prebiotic foods, but also another question in regards to butyrate came up. Which one would you have experience as to which one is the best, the calcium magnesium butyrate or the sodium butyrate? Have you experienced or worked with both, or?
Yeah, I've had experience with several of them. There are quite a few different forms out there now, so it can be confusing. Research is mostly done on the salts, so like the calcium or sodium butyrates. That's far and away the most common in research. Tributyrin is another form that's been well studied. I know that, I don't know if you want me to mention specific company names or just the type, but Tributyrin is definitely a form of butyrate that's well studied. There are some companies that offer butyrate so one of the concerns is that if you're just taking butyrate as a capsule, it's broken down in the small intestine and released there and then absorbed. It's not really getting to the colon, which is where butyrate is normally produced. Research does seem to indicate that at least some does get to the colon. But there are other formulations where, for example, it's sort of, quote, complexed to prebiotics so that it's not really released and active until it gets to the colon, and the colon microbes digest the fiber and then release the butyrate. So there's different formulations out there, but as is typical, not much research supporting individual companies' products and their specific formulations, so it's difficult to say from that standpoint. Clinically, the ones that are more targeted towards the colon seem to produce fewer GI symptoms. So that's been, and also just butyrate itself for some people is unpleasant. It's got a bad smell or a bad taste for some patients. There's liquid formulations and there's capsules. So there's options depending on sort of what the preference is for a patient. So as with anything, sometimes you have to try different products for a patient and see what's going to work best for them.
So I have used, there's different butyrate products in VMS, and I tell my patients to don't touch it with their fingers. Just put it into the lid and then into the mouth, or use tweezers or a spoon or something. So just don't touch it, because then you have the smell the rest of the day. So if you don't touch it, then it seems that most of them tolerate it, like that it doesn't keep tasting like a bad quality fish oil or stuff like that. So yeah, that's just my experience with it. I can't say which one I can't say which one works the best at all. Again, I give it mainly just to see if it can like reduce some of the symptoms, and I only give it hopefully until we've got the flora sorted out, so that there's an own production of short chain fatty acids that's working again. And that's of course like there's other tests that do measure the short chain fatty acids, but it reflects kind of, if there's a bad flora, then there's very often a bad balance in the short chain fatty acids. So that's my points to it. Then I don't know anything about this, but salicylate's role on the microbes in regards to mast cell activation syndrome. Do you have any comments to that?
Unfortunately, no, that's not an area that I'm familiar with. So I haven't specifically come across studies, and it hasn't really come up clinically for me. So I don't really, that's not something I can really speak to, unfortunately.
Super, no problem at all. So then I think let's move on to the case, and then I'll see if there's other things, and then we can take maybe more questions at the end. Okay.
It's a fairly quick case, so it should only take a couple of minutes or so. So this is a typical example, 43-year-old female. Just kind of a side note, this is not a full case study. Most of the case examples that I present in presentations come from the consultations that I do with Diagnostic Solutions Lab. These are 30-minute free consultations. So clinicians will just present a brief synopsis of their patient. So I don't have the full information. Typically, I don't have the follow-up either. So I don't always, unless they do a retest. So in some cases, I do see how their treatment affected the results. So I do have that if you have questions about what seems to work well for certain organisms. But this, again, is a 43-year-old female. Presented with abdominal gas, bloating, and pain, tends to increase throughout the day. Symptoms were associated with meals, but so far the patient had not been able to identify a specific food that seemed to trigger the symptoms, and the clinician had not yet run any sort of food sensitivity type testing. So the first test to run with the patient was the GI-MAP. We didn't see any bacterial pathogens. That page was clear. No parasitic pathogens. No viral pathogens. H. pylori was present, but it's at this sort of common low level of E2. We get a lot of questions about E2 levels of H. pylori. Is that something that needs to be treated or not? And since we can't really give direct medical advice, we can give information. And so E2 levels are very common. About 80% of patients who run GI-MAP have this. The majority of them do not seem to have any significant issues from this level of H. pylori, but a subset do. So that's where the kind of the gray area and clinical judgment comes into play. So you really have to determine, is this really consistent with symptoms, or is there likely something else that we see that's more likely to be contributing to symptoms? Because H. pylori really, by far, its most significant impact tends to be on stomach acid. So it's potentially, even at this level, having a negative effect on stomach acid. May not be significant for most patients in terms of symptoms, but if they do have a lot of GI symptoms and you see a pattern of overgrowth, maybe something to consider. So just anecdotally, some clinicians are more likely to treat H. pylori. They have mentioned that in their practice, even when they treat it at the E2 level, patients will often improve. So it's kind of an ongoing debate as to whether it should be treated or not. When I mentioned treat, in these scenarios where it's not a serious infection, it's not causing ulcers, of course, they're not generally going to be using antibiotics, but they will typically use herbal treatments, particularly mastic gum type formulas. In the normal bacteria section, this is, I think, the case I showed earlier when I was showing the example of low normal bacteria. So you can see there's a broad deficiency overall, including at the phylum level. The only one that was going the opposite direction is Akkermansia. We do see that elevated quite frequently on GI-MAP. That one we're actually likely to adjust the reference range in the near future now that we're getting some more data to analyze. But the cutoff level currently is at 5E4, which you can see over here on the right. We really don't see too many issues in the E4 range. So say if it's 8E4, a little bit higher than that, flagged as high, that's really just sort of higher normal. It's not really, doesn't seem to be much of an issue. But once it's in the E6 range, then we do tend to see that potentially correlating with other things. Most commonly with GI-MAP, high Akkermansia correlates with poor digestion. And that may be related to research showing that patients consuming a high sugar diet tend to have higher Akkermansia in some cases. So we do know that Akkermansia primarily thrives on the sugars from mucus. So that's why it's breaking down the mucus. But if you have excess sugars, which normally should just be absorbed in the small intestine, but if you have malabsorption, for example, or just consuming too much, you get spillover into the colon, or just generally too many carbs are getting in the colon, microbes are breaking down the fibers and releasing sugars. It can be a sign of just basically poor digestion and increased overall carbohydrate fermentation. And so it is commonly consistent with what we see on page 3. So this is a typical pattern for patients that have either IBS type symptoms, food sensitivities, SIBO type symptoms, etc. So bacillus is pretty commonly elevated. That one is really not regarded as, it's sort of debatable whether it belongs on the opportunistic page, but it is often overgrown. So we include it as kind of the overgrowth pattern, even though in and of itself, it's not really linked to a lot of issues or mast cell activation, etc. But Pseudomonas, on the other hand, we do often see that in patients that are symptomatic and have food sensitivities. Definitely, we talked about Staph aureus. Once again, patients are more likely to have those symptoms when you see a combination of these microbes. And then there's the high strep as well. In the autoimmune triggers section, Citrobacter was kind of getting up there. That might be notable. It's in the E6 range, but not officially high. That is one of those LPS and potentially histamine producing bacteria. So it might be another contributor. Whereas the Prevotella and Fusobacterium at these levels are normal in everyone. So those two are kind of the exception in this category, that they're always present and we don't really pay much attention until they're high. Candida at the genus level was high, so that's basically indicating a non-Candida albicans Candida species, excuse me, is present. So we know that that can stimulate mast cells potentially and contribute to this issue. No parasites were detected, either protozoa or worms. And then under the intestinal health section, this is a little bit of a confounder for some clinicians where if we see that overgrowth pattern that suggests reduced digestion, reduced stomach acid, but then they get to the digestion section, everything looks fine. It's good to remember that, of course, digestion has many steps, many aspects to it. We're only looking at a couple here in the digestion section. That's fat malabsorption with steatocrit. That looks good. We're also looking at pancreatic enzyme production. In this case, that looks good as well. So it does not appear that this patient has any issues with fat absorption or pancreatic function, but may still have either low stomach acid or they may have inflammation or dysbiosis in the small intestine. If you look back at page 3, we do know that Pseudomonas primarily lives in the small intestine, potentially can cause inflammation. Staph and strep are more dominant in the small intestine than the colon. Also may promote immune cells like mast cells as well. Of course, we talked about Candida. Most commonly, Candida is overgrown in the small intestine. So if you see these types of patterns, it's not directly telling you that patients have low brush border enzyme activity, but they could, because this is indicating likely dysbiosis and possibly some low-grade inflammation in the small intestine. And in research, that's widely recognized as being much more common than was originally thought. So it would be suspect, but there's no way for us to tell for sure. Now, if we look at the intestinal health section and the other markers, secretory IgA was low, most likely once again due to the lack of normal bacteria. Anti-gliadin was elevated, telling us that there is some immune reactivity, at least to gluten or to those cross-reactive foods. Again, we know there's a link with, actually I passed that one, with Pseudomonas, so not really much of a surprise that we see some gluten reactivity. And then of course zonulin in this patient was elevated, so telling us that there's also leaky gut that can contribute to this scenario. So as a summary, definitely H. pylori was detected, but it's low, may or may not really benefit from treatment. Low normal bacteria, so some intensive support for normal bacteria is typically warranted. High opportunist, in this scenario, clinicians are more likely to use antimicrobial herbs, usually kind of a broad spectrum approach, plus some targeted candida, anti-candida support with things like Saccharomyces boulardii, caprylic acid, etc. Low secretory IgA, in addition to supporting the beneficial bacteria, clinicians often will include colostrum or other types of immunoglobulins, glutamine, Saccharomyces boulardii, etc. Of course, with high zonulin suggests general leaky gut support. Anti-gliadin may suggest doing more extensive food sensitivity testing, or at least a trial of eliminating gluten or the related cross-reactive foods. So again, hopefully you can see from all this information in the case example that it can be quite helpful to identify some of these underlying imbalances in patients, and then addressing them may help to reduce their sensitivities. And we do actually see that play out in clinical practice. So thank you so much for listening, and feel free to check out our website if you want to look at more of our educational information. We have quite a bit there in terms of recorded webinars, etc.
Super. Now, more questions came up, so you're not free to go yet. So
I'm going to answer them.
Kane Litham is very determined in asking about the reliability about the zonulin marker. So come on, let us have some insights in regards to the zonulin marker, because you can also measure zonulin in blood and stool, and then you can have a zonulin marker that's negative, but then you can still have an increased permeability as if you're measuring sugar molecules. So can you comment?
Exactly. So zonulin is really just one of many factors involved in the overall intestinal barrier. So it's more of a, when it is positive, when it is high, that is really, in our experience, highly correlating with patients that have lots of dysbiosis, they have low secretory IgA. So it fits the picture, and it's telling us that likely this patient does have significant barrier issues. But if it's negative, certainly that doesn't mean that they don't have any barrier issues. That's why we have an older webinar, I think from 2 or 3 years ago, going through how to look at GI-MAP to assess the intestinal barrier. So it's much more than just zonulin. So you want to look at the beneficial bacteria. We saw that slide where the beneficial bacteria support the barrier in many ways. Secretory IgA, mucus, sort of the epithelial repair, regeneration. So definitely if you look at the normal bacteria and they're low, especially if there's a broad deficiency, that's telling us likely the barrier is not healthy. And then there's the specific factors like secretory IgA. Even beta-glucuronidase can be an indirect marker for barrier issues, excuse me, because it's mostly kind of a general indicator of dysbiosis. So if there's a lot of inflammatory type microbe overgrowth, even the mast cell stimulating microbes and histamine microbes, both of those, mast cells and histamine, also are known to disrupt the barrier. So it's important to kind of learn those connections, because that will give you a lot more insights into the state of the barrier, because it's really, the barrier is complex. It's not all about just leaky gut.
I very often do both the zonulin, and then I do an intestinal permeability assay with the 5 sugar molecules that we have in the system, except for that reason that I know that zonulin is just one marker. But if I have a patient where we have to like be careful with money, then, and I do a GI-MAP, then I would add the zonulin, and if it's positive, then I have something we can work with already there. If it's negative, then I say, well, there still may be something and these patterns, and then we could move into follow up with an intestinal permeability assay later on, or depending on what the patient says. So that's kind of what I do with, because we have different tools and than just the stool test. So just nudge so that you know that. What was the other one that came up? Yeah, then there's some questions about secretory IgA, and then there's another colleague answering in the group that a very high secretory IgA is probably because of an infection. And then we had a question coming up, but so if you have someone with low secretory IgA, you might want to use like immunoglobulins and treat them with that. And now I'm just going to ask you, because we couldn't answer that in the group, but can you give immunoglobulins to someone with a secretory IgA of, let's say, 6,000, like Christine is putting in here? Do you have any experience with that?
Yeah, I mean, it's not 100% clear whether immunoglobulins would be helpful or potentially not helpful when secretory IgA is already high. So you can kind of think of it this way, that oftentimes the baseline for many patients, and we see this really frequently on GI-MAP, that they'll have a low secretory IgA. So that's telling us that that's sort of their baseline. At that time, they don't appear to be necessarily reacting broadly to foods or to microbes, because then you would see a high secretory IgA. So if that's sort of the underlying issue, is that they generally have low function, the overall immune barrier is not healthy, but then you occasionally have a high level because they're reacting to something. That's usually more of a short-term thing. If you were to retest in a month or two, they may be back down to either normal or even low levels. So that's just kind of giving you a little bit of background information on the potential scenario for how patients may have high levels but may still benefit from general immunoglobulins. So immunoglobulins do have antimicrobial activity. So if you have lots of opportunistic overgrowth, certainly they are shown in research to help potentially reduce those opportunists. Good information on H. pylori, for example. And then they have other beneficial effects on the intestinal barrier. So I know of no case, sort of anecdotally or from research, where giving immunoglobulins when secretory IgA is high causes a bad reaction. It's theoretically possible, but I just haven't heard of that happening.
That's of course the most important, that it's not completely contraindicative in case we do that. Then Afifa says, can you mention betaine HCl, please? I'm not quite sure, Afifa, if you can just what is it you want to know about betaine HCl? I guess because I just listened to another podcast the other day where giving betaine HCl to control mast cell and histamine reactions in the stomach was recommended, but you'd mentioned that a little bit. So regarding low stomach acid and treatment of H. pylori and betaine HCl.
So we get a lot of questions about H. pylori in general, but also about whether or not it's okay to take betaine HCl when patients have H. pylori. The concern is mostly when H. pylori is high, and that's, of course, especially when there's documented significant gastritis, usually from an endoscopy. So if it's kind of just slightly elevated and it's mostly just having a negative effect on stomach acid, some clinicians do use supplemental acid. They generally don't report that patients have significant, issues from supplementing with acid. As always, the concern is really around the extent of their gastritis. And so there is some correlation with the levels of H. pylori. Generally, if it's only in the E2 range, much less likely to have significant gastritis. So then the acid is less likely to irritate the stomach. So that's, to my knowledge, that's the main concern, is patients can have pain if they have significant gastritis. That's going to erode the protective mucus layer and just overall make them more susceptible to the acid irritating the lining of the stomach. And of course, in severe cases, you would never want to do that when there's ulcers, et cetera. So that part is obvious, but it's not something that's officially contraindicated in patients that have detectable H. pylori. It may still be helpful, and lots of clinicians report that supplemental acid, when H. pylori is present but low, does benefit symptoms.
So how can you say, a protocol could be to, whether there's H. pylori or not, but at least you might have to treat some overgrowth dysbiosis anyway and give some antimicrobial herbs or compounds, and then at the same time maybe give something that has a healing and calming effect on the inflammation and irritation of the stomach lining. So you kind of kill what may irritate and try and heal before you start giving acid, because it would be like pouring acid into the wound, and that for some is very uncomfortable, where for others they don't necessarily feel it like that. But that's kind of the approach that I tend to I lower the bad guys, and then I try and heal at the same time before I start playing around too much with betaine HCl. But if I can see that the elastase marker is low, I really want to start playing around with betaine HCl as fast as possible. So I think that's sorry if I've missed something, or that. Oh yeah, there was something with Pseudomonas, and I must have been sleeping in that, because it was clicking up and down a little bit. Because if Pseudomonas is high but antigliadin IgA is okay, would you still consider gluten degradation? Please help me out here. I don't know what it means, the question. I didn't listen.
Yeah. So the antigliadin is kind of a long established marker. That's when it's positive. It's indicating there's likely gluten reactivity, except that we also know that antibodies cross-react. So there's always a possibility that it's maybe something else. Dairy protein, casein in dairy, can also cross-react. So high antigliadin isn't always due to gluten. But also, if the antigliadin looks fine and patients still seem to be reactive to gluten, there are broader gluten reactivity or wheat reactivity panels out there. And then also, if they have issues with wheat, it's pretty well documented now that they could have an issue with fructans. So wheat is a source of fructans that can cause GI upset. It's a source of these antitrypsin inhibitors, I think is what they're called, ATIs, that can cause intestinal barrier issues and inflammation. So wheat itself has many different components. So if they have a wheat issue, it may not be reflected in a high IgA. So it's more that when it is high, it is telling us there's likely some immune reactivity to gluten or to cross-reactive foods. But it's kind of like zonulin. If it's negative, it doesn't rule out that they might still have gluten or wheat issues.
And I will share the interpretation guide that belongs to this test with you when I send out your slides that you need to share with me, and then and the recording to you all as well. So yeah, I'll try and send out some additional information so that you have a neat email with lots of goodie information in that. So I think, Tom, let's let you have the time off now, so you can go out and play. And thank you very much for your presentation. As always, it's so nice to dig into a test and so thoroughly and get the references and research related to it at the same time.
Thank you. Thank you for having me today.
And I will invite you again. You know that.
Great. Looking forward to it.
Okay. Take care, everyone out there. Good to see you all. Thank you for chatting
Bye-bye.
to each other, answering questions that each other had, and so on. See you in a month or two's time when we have the next invites coming out. Bye.
If you do not already have an account with us, please contact our team and we will assist you. If you found this webinar helpful, please like and subscribe to our channel.